Hello, everyone. My name is Anastasia Canavariotti, for short Tessie, and I'd like to thank the organizing committee for accepting my abstract. My co-workers and I from Yenos Analytical in California and our collaborator, Dr. William Jack from New England Biolabs, have developed a point-of-care suitable assay for the detection and quantification of microRNAs from biological fluids. MicroRNAs or miRNAs are about 22 nucleotide long. They are about 2,300 human microRNAs known to regulate the post-transcriptional expression of messenger RNAs, and in this manner affect both physiological and disease pathways. Interestingly, they are found to be stable in biological fluids. Over 120,000 peer-reviewed publications at this time suggest that the upregulation or downregulation of a certain microRNA is associated with a certain disease and its severity. Thus, a small number of miRNAs could be used as a biomarker panel for practically any known human disease. Technical issues have resulted in conflicting studies and hindered the implementation of microRNA panels for the diagnosis of disease. miRNA levels in blood are reported to be in the range of 9,000 to 130,000 copies per microliter and believed to be lower in other biofluids. The range itself is modest, but corresponds to about 15 to 250 10 to the minus 15 molar femtomolar concentration, which is about 1 billion to dilute for most analytical tools. Most current techniques to quantify miRNAs depend on library preparation, PCR amplification, and next generation sequencing, with protocols that are lengthy, complex, prone to error, require skilled personnel and expensive infrastructure. Current technologies do not lend themselves for point-of-care assays. The last 30 years have seen breakthroughs in using nanopores as biosensors, and experimental nanopore platforms were shown to detect microRNAs. A single company, Oxford Nanopore Technologies, ONT, produces a device, the miNION, with a flow cell that holds an array of over 2,000 nanopores. The miNION is used for sequencing long DNA and RNA, but cannot identify or sequence very short RNAs such as the miRNAs. Our contribution to the field is that we have repurposed the miNION to do ion conductance experiments and have illustrated miRNA detection and quantification. Yenos miRNA detection combines two validated technologies. The first refers to ion conductance measurements via nanopores wide enough for the translocation of single-stranded nucleic acids, but not wide enough for the translocation of double-stranded ones. On the left is a representation of the alpha hemolysin nanopore embedded in a lipid bilayer, which is immersed in a high electrolyte aqueous solution, so-called buffer. Once voltage is applied on the system, ions flow only through the nanopore because of the presence of the insulating membrane. A single-stranded DNA, represented here by the orange string, also traverses the pore. On the right side, there is a simpler picture of the nanopore platform. During an ion conductance experiment, one measures the flow of ions during the nanopore, which is the ion current as a function of time. The constant flow of buffer ions via the pore, identified here as I0, the solid dark line that you see, is interrupted by the intermittent flow of other molecules such as the DNA, and these translocations appear as spikes or events with a certain dwell time t and at reduced ion current IR. The second part of our technology refers to labeling nucleic acids with a 1-to-1 mixture of osmium tetroxide and 2,2' bipyridine, called OSBP. This label was discovered in the 1960s. It adds from the top or from the bottom of the C5, C6 double bond of the pyrimidines and does not react with the purines. It does not cleave the phosphodiester bond and has a high affinity for thymidine. We have optimized this reaction called osmylation, use OSBP at a few millimolar at 25 degrees Celsius, developed conditions for selective thymidine labeling, and developed purification and analytical methods to monitor the extent of labeling. OSBP is a perfect nucleic acid label. Manufacturing and purification are safe and take less than an hour. Osmylated nucleic acids are stable in water, even at room temperature. As seen from the chemical structure of OSBP, this label is bulky, a feature we exploit. The picture to the left shows an osmylated RNA strand translocating via the ONT CSGG nanopore and illustrates that OSBP moieties extend parallel to the backbone and therefore do not significantly increase the radius of the single strand. The heart of our technology are the proprietary probes, which have a general and tested design. Excuse me. Each target microRNA has a unique probe, which is the exact complementary deoxyoligo. The thymidine bases in the probe are replaced by uridines, and the 5' end of the probe is extended by 3 adjacent thymidines. Upon osmylation, it is only the 3 extra thymidines that react with OSBP, and this results to strong hybridization between target and probe, but also a strong signal for the nanopore experiment. Due to the OSBP bulkiness, osmylated single-stranded nucleic acids traverse the ONT nanopores about 1,000 times slower compared to the native ones. Because the ONT data acquisition rate, which is 3 data points per millisecond, is relatively slow, native nucleic acids are not detected. There are no events, whereas osmylated nucleic acids produce events. The concept is shown in the figure. When the test sample is in the flow cell and voltage is applied, all the nucleic acids approach the nanopore. Single-stranded nucleic acids, including miRNAs, traverse the nanopore but are not detected. A Yenos probe will traverse and produce an event. But in the presence of its target microRNA, the probe will hybridize, will not traverse the pore, and thus it will be silenced. Nanopores remain active even in the presence of hybrids because the electronics reverses the voltage and the pores are freed. Our protocol uses 0.2 ml blood sample, 4 drops, treated using a commercial kit for total RNA isolation from blood, such as the Monarch from New England Biolabs or the QAGEN kit. 100 microliters of pure total RNA is isolated in water and aliquoted into 20 microcentrifuge tubes. To each tube, one adds 80 microliter of ONT buffer and 5 microliter of a unique microRNA probe at a known concentration. Each sample is loaded into a MinION flow cell device, and the device connected to a laptop loaded with the ONT software to run the experiment. See the middle figure. Actually, we're using the same software as for a sequencing experiment. We acquire data, figure to the right, for 60 minutes, then analyze the raw data by our publicly available OSBP detect algorithm that reports counts of events using user-selected parameters for residual ion and dwell time. 4 consecutive experiments are shown here with materials at 10 femtomolar concentration each in the flow cell. The gray dashed line, see to the left, is from a test sample or buffer. The dark green trace is a test sample with microRNA 21 and its corresponding probe. The light green trace is a sample with microRNA 140 and its corresponding probe. Both these samples exhibit counts comparable to the ONT buffer, meaning the probes are silenced. In contrast, the red trace, easier to see on the right figure, comes from an experiment with microRNA 140 target and a probe for microRNA 21. Because there is no complementarity in this set, the probe is not silenced and exhibits a remarkably large count of events. The quantity of the target is deduced from the known quantity of the added probe based on a one-to-one hybridization. We are currently working on reducing the noise, quantifying the probe signal, and multiplexing probes for detection of up to 4 microRNA targets per single experiment. Last but not least, we thank you for your attention.